The vast majority of cancer-related fatalities are not caused by the initial tumor, the one you can point to on a scan. That primary mass is often manageable. The real danger comes from its children, the microscopic colonists it sends out to establish distant settlements in the lungs, liver, or brain. This process, metastasis, was long viewed as a grim, almost inevitable final act in cancer's playbook. But in the late 1980s, a team led by Patricia Stieg, studying intensely aggressive melanoma cells, stumbled upon a profound anomaly. When they injected these cells into mice, some simply failed to spread. They grew into tumors, certainly, but they stayed put, respecting their original boundaries. It was as if they had forgotten how to be invasive. Peering inside these curiously well-behaved cells, researchers found a particular gene working overtime. They named it N23 for non-metastatic clone 23. It wasn't a gene that promoted growth or killed the cell. It was something entirely different. It was a molecular supervisor, actively suppressing the wanderlust that makes cancer so deadly. It was a brake pedal built right into the cellular machine. To understand how a brake works, you first have to appreciate the vehicle's impulse to move. A healthy tissue, or even an early-stage tumor, is not just a pile of cells. It's a tightly packed, well-organized neighborhood with strict zoning laws. Cells are physically bound to one another and to their underlying support structure, the extracellular matrix, by a series of molecular rivets and glues. A key one is E-coherin, a protein that acts like cellular Velcro, ensuring everyone knows their place and stays connected to the community. For a cell to metastasize, it must first perform a remarkable act of social and biological rebellion. It must dissolve this glue, retract its connections, and change its very shape from a stationary, predictable epithelial cell into something more fluid, predatory, and mobile, a mesenchymal cell. This profound transformation is known as the epithelial-mesenchymal transition, or EMT. The cell becomes a fugitive, shedding its old identity to survive a perilous journey through the bloodstream or lymphatic system. So where does our brake pedal, the N23 protein, intervene in this dramatic escape plan? It doesn't appear to work by simply reinforcing the cellular glue or building higher walls. Its mechanism is far more subtle and far more fundamental. N23 is an enzyme, a nucleoside diphosphate kinase, or NDPK. That's a technical term, but its job is elegantly simple. It's a currency exchange for cellular energy. It expertly converts molecules like GDP into GTP and ADP into ATP. These molecules, especially GTP, are the specific currency required to power the dynamic restructuring of the cell's internal skeleton, the cytoskeleton. Escaping a tumor crawling, changing shape, invading tissues requires a massive amount of precisely directed energy. N23 acts like a stingy but wise energy accountant. By controlling the local supply of GTP, it prevents the cell from powering up the very micro-machines it needs to dismantle its home and begin its journey. The engine can't start if the fuel pump is off. But a cell's decision to move isn't just about internal energy. It's about external information. The cell is constantly listening to signals from its environment. Imagine molecular messengers, growth factors, and other signaling proteins constantly knocking on the cell's outer surface. Some of these signals say "stay and grow," while others whisper enticingly, "The environment elsewhere is better. It's time to go." A cell brings these messages inside through a process called endocytosis, swallowing them in tiny membrane bubbles for processing. Astonishingly, N23 seems to be a key regulator here as well. It interacts with and controls the activity of proteins like dynamin, the molecule that acts like a pair of scissors, pinching off those message-filled bubbles from the cell surface. By fine-tuning the rate of this process, N23 effectively manages the cell's mailroom. It doesn't block all messages, but it can prevent the cell from becoming overwhelmed by and overreacting to the very signals that would encourage it to pack up and leave its neighborhood. This raises a crucial, almost philosophical question. If our cells are equipped with such an elegant, multi-layered defense system against their own worst impulses, why does metastasis happen at all? The answer is a stark lesson in the brutal logic of evolution, playing out on a microscopic stage inside a single body. A tumor is never a single, uniform entity. It is a chaotic, thriving metropolis of trillions of competing cells, each with slight genetic variations from constant, imperfect division. In this hyper-competitive environment, any advantage, no matter how small, leads to dominance. All it takes is for one of those trillions of cells to randomly acquire a mutation that silences its N23 gene or the pathway that reads it. In that one cell, the brake pedal is suddenly gone. The energy accountant is fired. The mailroom security is disabled. That cell and its descendants now have a profound survival advantage. Cancer's spread, then, isn't an inevitable destiny written into its fundamental code. It is the tragic outcome of a numbers game, a relentless, microscopic war of attrition where the body's sophisticated defenses have to win every single time, but the rebellion only needs to win once. Monotony has grown fast. Nearly 2,700 people have watched in the last few weeks, yet less than 0.03% are subscribed. It would mean a lot if you joined by hitting subscribe. And again, thanks for watching.